Live.
Good evening, everyone. So today we are starting our biology series. This program is designed as an outreach to non-biologists and inspire them to think cellular and molecular when the policymakers, patient groups, Ayush trainee, other disciplines, and lay public approach their field, disease, integrative health, education paradigm, or when they go about your lives confronted by terms such as antibody, COVID, conjunctivitis, retina, AMD, vaccine, etc. Another objective of this series is to highlight the biological phenomena underlying disease and wellness. I hope this interaction series between global science leaders and the field and postdocs represent two knowledge levels, will arouse your interest in the biology research and its funding and catalyze the dialogue between science and society and communication. I'm pleased to welcome today's speaker, Dr. Shweta Modgil, and commentator for the today's session will be Dr. Anand Swarup. So Dr. Shweta Modgil is doing postdoc from Emory University, Georgia, USA. She has been involved in vision research for last 7 years, and currently she is a postdoctoral fellow in the School of Medicine, Emory University. Her main area of research interest involve retinal ischemia and traumatic eye injury. Her present work focuses on identifying new small molecule agonists to TRKB receptors to be used in prevention of retinal degeneration. Our moderator, Dr. Anand Swarup, is well known in research of retinal neurodegeneration, genetics of age-related molecular genetics, and design new therapeutic paradigm using cell, gene, and small molecular-based approaches. He is from National Eye Institute, NIH. He has trained over more than 500 students and has more than 350 publications. He has been awarded many awards, including he has been granted Professor P.N. Chutani Chair as Distinguished Medical Scientist at our institute during 2015 and 16. And he has also received the prestigious Alcon Award for Outstanding Vision Research in 2011. So he has 4 patents in his credits. So I hereby welcome Dr. Shweta to deliver her talk.
Thank you, Manjari. Let me share my screen. I hope you can see this now.
Yes.
Okay. Thank you everyone for joining us today. So I will be talking about structure of retina and process of phototransduction. So this is the brief layout of my presentation, what I will be talking about today. So I'll start with the gross anatomy of eye, followed by retinal layers, which includes retinal pigment epithelial, photoreceptors, outer plexiform layer, inner nuclear layer, inner plexiform layer, and then ganglion cell layer. And last, we will discuss about the phototransduction process. So starting with the basic introduction, eye is an extension of central nervous system as it is developed from the neural tube. And this is the only part of central nervous system that we can see from outside. So the main photosensitive part of eye is retina, and it is a multi-layer tissue comprising of different types of cells. So light is focused on the retina, where it is converted from light energy into chemical and finally into an electrical signal, which is sent to the brain for the decoding. So this is the gross anatomy of the eye. Towards your left-hand side in the front, on the top panel, you can see whenever you look into eye of a person, what you can find is a black hole at the center, which we call as a pupil, through which light enters. Then it is followed by a circular structure, which is called as iris. And it acts as a diaphragm, which controls the amount of light that is entering inside. And this is the part which gives eye its characteristic color. When we say that a person has a blue eye or black eye, that's because of the pigment that is present in the iris. This white portion that you see is a sclera, which is also the outermost layer of the eye, which we'll discuss later on. Towards the right-hand side, you can see a broader image of eye, where you can see that eye is composed of 3 basic structures. The outer structure is formed from sclera as well as cornea. This cornea you can see here because it is a transparent structure which covers your pupil as well as iris. So this cornea is in close contact with the sclera and it forms the outermost layer of the eye. This is followed by your intermediate layer, which is also called as uveal tract. And this basically is made up of three structures. From the innermost side, it is choroid, which basically provide the blood supply to the retina. And then there is this ciliary body, which helps in the suspension of the lens and its accommodation. Then it is followed by the iris, which I just said that it acts as a diaphragm and it controls the pupil dilation as well as constriction. Oh, of course, the innermost layer that is in red color, here you can see that is retina, which we'll discuss in detail. So retina is a multi-layer structure, and as you can see, it is composed of about 10 layers. Starting from the outermost side, it is pigment epithelial, followed by photoreceptor layer, then outer limiting and outer nuclear layer, followed by outer plexiform layer. So these four layers, you can see that these are formed by just one type of cell, which are called as photoreceptor. And we will discuss the detail of these photoreceptors in coming slides. Then is an inner nuclear layer, followed by inner plexiform and ganglion cell layers, which comprise of your retinal ganglion cells. The axons of the retinal ganglion cells then form nerve fiber layer, and these nerve fibers wrap together to form the optic nerve, which takes the signal to the brain. Those who are working in the retina are more familiar with this diagram on your right hand side, which you see under a microscope when you section eye. So we'll start with the retinal pigment epithelial. So this layer is the outermost layer of retina, which is hexagonal cells with a cobblestone morphology, and these are very tightly packed because of the tight junctions that are present within the cells. So this RPE has basically two domains. One is the basolateral domain and another is the apical domain. Basolateral domain is the domain that is towards the choroid, that is blood supply, whereas the apical domain is the domain of RPE, which is towards the photoreceptors. Now, retinal pigment epithelial perform various functions, and one of the function is the light absorption. Whatever extra light that is not absorbed by the photoreceptors, which is not actually sensed by the photoreceptor, are absorbed by the retinal pigment epithelial, so that it is prevented from being reflected back. And for this, the RPE has special organelles, which are called as melanosomes, which contains melanin, which help in the absorption of the extra light. Also, because of photooxidation that is taking place, there are various free radicals that are formed, and to combat that free radicals, there are antioxidants that are present inside the RPE. Further, because of the light, there is some increase in the temperature or heat is generated, which is carried away because of the high perfusion of the RPE. Next, blood retinal barrier is also another function that is performed by retinal pigment epithelial. As I just said, that there are tight junctions which are present in the cells, because of which the RPE is impermeable to any material. So whatever material is transferred between the blood supply as well as retina is only through the retinal pigment epithelial. So retinal pigment epithelial perform the main task of transporting the material to and from the blood supply to the retina. So nutrients such as glucose, omega-3 fatty acid, as well as retinol is transferred from blood to the retina, whereas from blood, from the retina, the waste materials such as water, lactic acid, as well as bicarbonates are transferred to the blood through RPE. And also the ions, they move both ways, either from blood to retina or from retina to blood to maintain the osmotic pressure. Next important function of retinal pigment epithelial is secretion. That is, besides just transport, they can secrete certain molecules which are responsible for the health of retina as well as the microvascular. These includes various neurotrophic factors such as FGF, VEGF, CNTF, PDGF, and TIMP. The next important function that RPE perform is its involvement in the visual cycle. So what happened is that photoreceptors are main light sensing cells in the retina, and for that they have pigment which is called as opsin and a chromophore that is 11-cis retinal. This 11-cis retinal is formed in the retinal pigment epithelial from the retinol, that is vitamin A, which is taken up by the RPE from the blood and converted into 11-cis retinal. This 11-cis retinal is then transported to the photoreceptor, where it helps in the light absorption. Now, once light falls onto the photoreceptor, this 11 cis-retinal is converted into 11 trans-retinal aldehyde. Now the photoreceptors, they don't have your isomerase enzyme. So this 11 trans-retinal aldehyde that is formed here cannot be converted back into 11 cis-retinal in the photoreceptor itself, because of which this transform is again transported back to the RPE for regeneration. And this regeneration cycle that takes place between the RPE as well as photoreceptor is called as visual cycle, which was first discovered by George Wald, and for which he also got a Nobel Prize in 1967. So in this diagram, you can see what happened is that this is your RPE cell, and this is the apical domain of RPE cell, which you can see here. And this is your photoreceptor cells with this outer segment containing the photopigment. So RPE cell, it contains gets the vitamin A from your blood supply and convert into its cis-retinal aldehyde form. This cis-retinal aldehyde form is transported to the photoreceptor, where it become an integral part of photopigment. Now, when light falls on this photoreceptor, this cis form is converted into your transretinal aldehyde form. This transretinal aldehyde form then further is transported from rod outer segment to the cytoplasm of the rods, which is this purple thing that you can see in the diagram. Now in the cytoplasm, this this transretinal aldehyde is converted into transretinal by the action of RDH. And this transretinal form is then transported from your cytoplasm of photoreceptor to the interphotoreceptor matrix, which is a space between your RPE and your photoreceptor. Here, this transretinol is carried by a carrier protein, which is IRBP, and transported to the retinal pigment epithelial. In retinal pigment epithelial, again, there is a carrier protein, which is called as CRBP. This is your cellular retinal binding protein. So here, this CRBP carry this transretinol to the enzyme complex, which further contains 3 enzymes. One is LRAT, another is RPE65, followed by RDH5. These 3 enzymes later convert your transretinol into the 11 cis-retinal aldehyde. So LRAT is performing your esterification, that is, it is converting your transretinol into transester. Now, once transester is formed, isomerase and converting your trans form into cis form. So here you have cis-retinol, and cis-retinol is further oxidized from with the help of RDH5 to the cis-retinal aldehyde, completing the cycle. Now, this cis-retinal aldehyde can again move back to the photoreceptor and became part of photopigment. So next important function that RBE perform is your phagocytosis. So what happened is that because the photoreceptors are continuously sensing the light, and because of which they undergo a constant destruction, and they just shed off their outer segments into the interplexiform, into the interphotoreceptor matrix. From here, these are taken up by the RPE with the help of 3 proteins, that is CD36, MERTK, and alpha beta 5 integrin, which help in the phagocytosis of this outer segment and regeneration of photoreceptors. So loss of MERTK, which is an enzyme involved in phagocytosis, it is known to lead to human retinal pigmentosa, which is a disease in which your photoreceptor loss takes place. RCS rats, which are a model for inherited retinal degeneration, also contains the loss of MERTK gene, which leads to retinal pigmentosa in these animals. Now, as I told you that RPE and photoreceptor, they lies in very close proximity to each other, and photoreceptor depends upon RPE for nutrition as well as their maintenance. So there is a very little space between photoreceptor and RPE, which we call as subretinal space. This space, you might have heard about the subretinal injection. This is the space where we administered most of the studies that are carried out using stem cells are administering stem cells into this space so that they can nicely integrate with the remaining of the retinal layers. Also, the implants that are such as Argus2 or IMS, alpha IMS, they are also integrated into the retina, into the subretinal space, so that they can work as a artificial photoreceptors. Now, sometimes what happens is that these photoreceptors are pulled away from the RPE, and this leads to what is called as retinal detachment, which leads to your hazy vision. or blurry vision. So age-related macular degeneration is another disease which results into central vision loss. And this is characterized basically by the drusen formation in the Brooks membrane, which is the region between RPE and choroid. Dr. Anand is basically a pioneer who is working in AMD, and he will talk much about this maybe in his slides or in his talk. So next we have photoreceptors, and photoreceptors are basically two types, rods and cones, and they have a structure which comprises of four different regions. Outer segment, which we just discussed, is contain stack of membranes and contain photopigment that is basically comprised of opsin as well as chromophore, that is vitamin A. Followed by there is an inner segment which contains your Golgi endoplasmic reticulum as well as mitochondria that provide energy for the cell. Then is your cell body, which contains your nucleus and a synaptic ending, which transmit the signal from here to the secondary order neurons. So here you can see the outer segment in which stack of membrane you can see, and this is just an enlarged view of the photopigment that is present in the disc membrane. This photopigment is actually a GPCR, that is G protein coupled receptor, because so it has a basic structure of GPCR, that is an extracellular domain followed by 7 transmembrane domains and then an intracellular domain. It is the 7th transmembrane domain where we have a chromophore attachment site, where a chromophore, that is 11-cis retinal aldehyde, attaches. So retinal pigmentosa, as we also discussed earlier, is a disease which lead to blindness due to loss of photoreceptors. Now coming to the next layer, which is outer plexiform layer. Here you can see this is nothing but it contains the synapses between your photoreceptor as well as secondary order neurons, which are lying in inner nuclear layer. Now what is present in the inner nuclear layer? So inner nuclear layer comprises of basically 3 to 4 types of cells. One are the bipolar cells, which connect your outer plexiform layer to inner plexiform layer. And in human, there are around 11 types of bipolar cells that have been identified. These are basically of 2 types depending upon the light response, either on bipolars or the con bipolars. So on bipolar, they become depolarized when light falls on the photoreceptors, whereas the off bipolars, they become depolarized or activate when the light is not falling on the photoreceptor or there is just no light present. So on bipolars and off bipolar cells are actually responsible for 2 separate channels of information flow. And this is because of these bipolars, we are able to see a dark background over the white contrast as well as white background over the darker objects. Next cells which are present in the inner nuclear are the horizontal cells, and these are basically laterally interconnecting neurons, which connects your rods with rods or rods with cones, and they are also responsible for inhibitory feedback to the rods and cones receptors, which results in lateral inhibition. Lateral inhibition is actually the activity by which the neurons are actually inhibiting their neighboring neurons from activating. So this results in high contrast of the image. Next in the nuclear layer are the amacrine cells, and there are various amacrine cells that have been recognized, like 40 different types of them are present. Basically, A2 amacrine cells are the main amacrine cells that help in the relay of signal from rod bipolar cells to the ganglion cells, because rod bipolar cells don't form a direct connection between the ganglion cells. So A2 amacrine cells are the main relay cells that transfer this information from on bipolars to the ganglion cells. Another amacrine cells are the starburst amacrine cells that play an important role in directional selectivity as well as directional motion. Then dopaminergic amacrine cells, they are known to release the dopamine and help in the light absorption, light adaptation and modulation of signal, as well as in their role in circadian rhythms. Last layer is the ganglion cell layer, which contains your retinal ganglion cells. And their axons are basically terminates into an optic nerve, which carries your signal to the lateral geniculate nucleus and superior colliculus. 20 different types of retinal ganglion cells have been identified, and they are broadly divided into 3 major types that are on RGCs, off, RGCs and on-off RGCs depending upon when they are getting activated, whether on the light, when there is light on or when there is light off, depending upon when they are activating. They are broadly divided into these three categories. So this is your inner plexiform layer, which contains connection between your inner nuclear layer as well as ganglion cell layer. It basically is divided into different stratas, A and B substrata, and A substrata contains your connections between your off bipolar cells as well as off ganglion cells, whereas your B substrata contains major signals from off on bipolar cells and the on ganglion cells. Beside this function, ganglion cells also function as a photoreceptors, subset of RGCs, which are called as intrinsically photosensitive RGCs, are known to be photosensitive. For example, OPN4 RGCs, which are also called as melanopsin, they are known to involve in regulating your circadian rhythm by sending signal to your suprachiasmatic nucleus, which is a master clock in your body. Then is the OPN5 RGCs, which are also called as neuropsin. They are known to regulate your local retinal clock. Beside the master clock, we also have a local clocks in various tissue. So OPN5 is known to regulate your local retinal clock. Now, glaucoma is a disease which is characterized by loss of retinal ganglion cells due to increased ocular pressure. And as we have talked about AMD, in which your central vision is lost, in glaucoma, you can see the center of the image very clearly, but your peripheral vision is hazy. Apart from neural cells, there are certain glial cells present in the retina, which includes your Muller cells, astrocytes, as well as microglia. So Muller cells are principal glial cell of the retina, and they are basically derived from the same retinal progenitor cells, which form your neural cells as well. So they are true glial cells of the retina, and they are also form your inner and they also play a role in the inner and outer retinal barrier. Then are your astrocytes that are not glial cells of the retinal neuroepithelial, but they enter the developing retina from the brain. So they are basically present near the optic nerve. Then are your microglia cells. They are mesodermal in origin retina with the retinal blood vessel development, and they are present ubiquitously in the retina, and that is almost in every layer, and perform the function of phagocytosis of the debris as well as dead neurons in the retina. Now coming to the phototransduction process. So before we start, we should know this, that photoreceptors are actually depolarized in the dark. And when light fall on them, they become hyperpolarized. So there is a dark current that is going on inside the photoreceptors because the sodium channels are open during the dark because of the cGMP ligand, because of the cGMP gated channels. But when there is light, when light fall on the photoreceptor, what happens is that the cGMP gated channel closes, because of which the flow of sodium channel inside the cell is disrupted, which lead to hyperpolarization of the photoreceptor. Because of hyperpolarization, no glutamate is secreted, or there is no synapse formation between the photoreceptor and the second order neurons, and the signal is actually not transmitted. So one thing to be remember in this case is that only 1% of channels are affected by light, and they decide the fate of photoreceptor, that whether they will become depolarized or hyperpolarized. In other words, we can say that 99% of channels are already closed in the dark, and light can only control the remaining 1% of the channels. Now, what is phototransduction? So this is the structure that we discussed earlier as well, that this is the outer segment containing the photopigment, which is a GPCR. So what happened is that light falls on this photopigment, and it is absorbed here. This light energy is then converted into your chemical energy, and finally into your electrical energy. So this process is called as phototransduction, which involves the receptor, which is rhodopsin, this one, and then a G protein, which is transducin, and there is an effector molecule, which is cGMP phosphodiesterase. So transduction has basically three subunits, that is alpha, beta, and gamma. When there is no light, this alpha, beta, and gamma subunits light together and form a complex. But when the light falls on the photoreceptor, phosphorylation of G protein take place. That is, this alpha, it become detached from the beta and gamma subunit, leading to the formation of alpha GTP, which is your activated G protein, activated G protein. Now, rod and cones have different isotypes of transducin, and this is really useful because different isotypes can be deleted from the cells, and which lead to the formation of, which lead us to create animal models in which either rods or cones are affected. For example, GNAT1 knockout or null mice contains dysfunctional rods, whereas
1 or 2
GNAT2,
minutes, Shweta.
sorry, yes, sir.
1 or 2 minutes more.
Okay. So GNAT2 null mice actually contains null mutation in transducin 2, and because of which there are dysfunctional cones, but rods are intact in these animals. So photodiesterase is another enzyme which is involved in phototransduction. And here there are alpha, beta, and again gamma subunits. And gamma subunit actually binds to the alpha, beta and render it unavailable for its activity. But when light falls on the photoreceptor, G protein is activated and PDE is also activated because your gamma subunit is detached from alpha and beta. So coming directly to the phototransduction process, because we don't have much time. So what happened is that when light fall on the photoreceptor, what happened is that this rhodopsin become activated, that is R become activated. This further activates your transducin protein, which is a G protein. This transducin after activation then further activate your phosphodiesterase by removing the gamma from alpha beta. This PDE, that is phosphodiesterase, become activated, and this further convert your cGMP, that is cyclic GMP, which is secondary messenger in this case, into your GMP. Now, once cGMP is converted into GMP, these channels, which are cGMP gated channel, became closed. Now, when this closes, the flow of sodium inside the photoreceptor is halted. Because of this channel, there is hyperpolarization of photoreceptor and glutamate is not secreted. So how the signal terminates is that this G protein, which is activated here, this G protein which is activated, this become inactivated because G protein has an intrinsic GTPase activities, because of which this G activated transducin is rendered inactivated again, and gap is a complex which help in hydrolysis of GTP to the GDP again. This when this G alpha GDP dissociates from PDEV, this again allow this gamma subunit of PDE to bind to alpha and beta again and inhibit the PDE. Also, your rhodopsin, which has been activated earlier, that become phosphorylated by the activity of rhodopsin kinase, and again your rhodopsin became inactivated. So the phototransduction became terminated in this case. So summarizing, as we see that our eye consists of three structures, basically three layers: outer layer containing sclera and cornea, followed by middle layer, which is containing the uveal tract, that is your choroid, ciliary body, as well as iris. Then is your retina, which consists of your outer layer, that is retinal pigment epithelial, followed by photoreceptors. Then is your inner nuclear layer, comprises of your amacrine, horizontal and bipolar cells. The connections between your photoreceptor and inner nuclear layer is called as your outer plexiform layer. Then is your ganglion cell layer, and the connection between ganglion and inner nuclear cells are called as inner plexiform layer. Ganglion cell layers form your axons of ganglion cell form your nerve fiber layer, which later on encapsulate together to form your optic nerve, which carry the signal to the brain. So this is all, I guess. These are the references for my presentation. And at last, I would like to thank Dr. Akshay for giving me this opportunity, as well as Dr. Anand. I mean, he's so modest to be here for this basic presentation. And thank you everyone for listening.
Unshare your slide. Manjari, you can tell her to unshare the slides. Yes.
So great, Shweta. That was a very nice introduction to retinal structure and function, and sort of you basically summarized how we have, how we initiate the process of vision, and how that, whatever we see is then transmitted to brain for further analysis. So that was very good. What I'm going to do is uh, I'm going to share my screen and give you a little bit more about the first sort of layer of neurons, the photoreceptors. Uh, and my lab actually works on all aspects of retinal biology and disease. Let me first share my screen and see where that is. Where is my slides? Okay. All right. Can you see my screen?
Yes.
Okay. All right. So after that excellent introduction, what I'm going to do is I'm going to talk to you about uh, the sensory neurons called photoreceptors. And I have been fascinated, or you can say even obsessed by these cells for last 30 plus years. Um, they are the cells that initiate vision. If you have no photoreceptors, you are not going to be able to see. So we focus on many different aspects of retinal biology, retinal disease, and therapy, but the primary focus in general is on photoreceptors. So these are various individuals in my lab, and which is called NNRL, which is Neurobiology, Neurodegeneration and Repair Laboratory at National Eye Institute. So just to add to what Shweta was saying, retinal photoreceptors initiate the vision process, but I think the key part is that these are post-mitotic cells. Once they are born, they are not going to divide again. They are not going to regenerate on their own. They are not going, it's very, very hard to sort of replace them. So it's very important that you keep these photoreceptors alive and functional. Uh, so how does our retina keep these cells functional for almost 80, 90, 100 years of our life? And that's because of these multiple unique features. One of the features is what I think Shweta alluded to, is this we have these outer segments, which there are a lot of these outer segment discs in these, which are shed every day. 10% of these discs are shed every day and phagocytosed by retinal pigment epithelium. That sort of keeps these cells young, right? So if 10% of these discs which capture light are renewed every day, means in 10 days you have this whole functional part of the cell is replaced. So rather than replacing the whole cell in the retina, what happens is that you renew the things that are getting damaged with light. Okay, so these are rod photoreceptors and these are cone photoreceptors as depicted here. Rod photoreceptors, I must mention, allow you to see in dim light, whereas cones are more important in humans because of their unique role in daylight, in color vision, and similar processes. And they also give you very high visual acuity. That means you can resolve very minute structures. And I think Shweta alluded that much of that happens later in the inner retina. These cells, during phototransduction, as was mentioned earlier, they hyperpolarize. But during the dark state, these cells, when they are depolarized, they consume a lot more energy than actually in light. To keep those channels open, you require very high amount of ATP, and that will require very high metabolic activity, very high consumption of ATP and all that. So another very unique feature of these photoreceptors is that their synaptic transmission is very different than most other neurons. And these have ribbon synapses, which allow them to release glutamate in at very fast rate. Now, as I mentioned earlier, if these cells become dysfunctional or they die, you lose your photoreceptors and you will not be able to see. So how do we understand, how do we try to uh, design different therapies for retinal and macular diseases? So here you are. Let me give you a few very basic concepts, and that's all I'm going to try to do today in the next 5 or so minutes. In general, when you say somebody has got a disease, what is disease? Disease is caused by changes in the normal behavior of a cell or as a whole, as an organism. Okay, so if photoreceptors, if their function is perturbed, you will have disease. Now, disease actually happens when the function is perturbed so much that you are not able to maintain the normal behavior, and then you'll have to go to a doctor or something and say, okay, you know, I'm not able to see. So how does that happen? That can happen due to genetic mutations, changes in our genome, or it can happen because of environment, because of UV, because of aging, because of many nutrition actually also has a major impact. And since this is for yoga scholars, I would also mention that exercise, yoga, and all other your daily routines also have an impact on cellular functions, including the photoreceptors. So if you want to understand disease or what is abnormal, you need to know what is normal. So my lab has got multiple different projects. One of the key part of our work is to understand healthy phenotypes, how different cells, specifically photoreceptors, are generated in the retina, and how they keep functioning throughout your life, and what happens when you age. Then we also want to understand the interaction of these photoreceptors with the environment in response to, for example, nutrition and exercise. And then on the other hand, we want to understand how different genes and genetic variants can impact these healthy phenotypes. And whenever there is a perturbation because of environmental changes or genetic variations, you get disease. And we focus on all of different aspects. And this understanding of these basic fundamental pathways allows us to design different kinds of therapies. So one of the first thing that we have been doing for a very long time is trying to understand how photoreceptor develop from retinal progenitors or even actually stem cells, pluripotent stem cells. There's a very complex process that even after these photoreceptors sort of are born, that means these cells exit cell cycle, there's a whole process of morphogenesis. These unique photoreceptors have photoreceptors very unique morphology, as I mentioned to you earlier, from outer segment to synaptic terminals. And how does that happen? So the transcription factor, my lab discovered a while back, the transcription factor that is key to this morphogenesis and so to this self-determination morphogenesis is NRL, or neural retina leucine zipper gene, and along with another transcription factor called thyroid hormone receptor beta, and multiple other very important proteins like cone rod, homeobox, CRX, and other, they all work together to generate specific gene expression patterns that give their morphology as well as function. So NRL, if you knock it out in mice, you have no rod photoreceptors and they all become short wavelength cones. And now you put them in cone at an appropriate stage in differentiation, all those cones can become rod. So NRL can determine the fate of rods and also change the fate of cones, short wavelength cones to rods. Now, understanding the genome requires you to have very good understanding, rather, understanding of the disease requires you to have good understanding of the genome or genetics as well, because our genome of 3 billion nucleotides uh, has over 100 million variations. Each of one of us carry tens of thousands of variants which are unique to us. They allow us to have very unique facial features, unique functions, and the individual that we are, the individuality that we have is really determined by these genetic variation. Yet, even though all of these cells have same DNA in our body, yet each cell has very unique phenotype and function. How does that happen? We would like to understand that in the context of photoreceptors. And that requires a big systems level approach. That means you need to do genetics, you need to do biochemistry, you need to do computational biology, you need to do cell biology and physiology, as well as basically you need to integrate a lot of different kinds of studies in order to understand or to answer a question. And that's what we are trying to do. And we try to do that in the context of photoreceptor, photoreceptor function, photoreceptor disease. And for example, in case of inherited retinal degeneration, which is generally Mendelian, what that means is that defect in a single gene can cause the disease or change in phenotype. We want to understand using mouse models or retinal organoid models how the disease is caused and how we can rescue that. In age-related macular degeneration, it is very complex where the genetic variants have to interact with environment, and it's also an age-dependent process. So aging is very important factor. And again, that's why we try to understand the role of genetics, the role of environment, as well as the role of aging in causing age-related macular degeneration. So there are many different ways for treatment that are being designed all over the world. One is this, what we call gene-based therapy. It has been very successful, at least in one case, where in patients with LCA or Leber corneal amaurosis caused by defects in RP65, one can now replace the gene, and these individuals are able to then have functional vision. And it does work. There are some issues later in life, but that is still being worked out. How successful that will be is still something that we need to think about. The problem with gene therapy is that it's very expensive. It takes a lot of time, and for each gene and mutation or set of mutations, it needs to be designed separately. Right now, the therapy for RP65 costs close to $1 million. How many people all over the world, and particularly places like in India or even in US, it's not that simple. Cell-based therapy, that's why gene-independent therapies are very important, and one of these is stem cell-based therapies. Again, there have been some issues. My lab, along with, in collaboration with Robin Ali, showed a while back that photoreceptor replacement might be possible. However, some later experiments showed that some of those studies still require extensive investigations because of the same transfer of material between host and donor cells. So a better strategy might be to keep whatever we have alive. And that's where nutrition and yoga and environment, which I was trying to mention earlier, could be very important. Rather than trying to treat the cells at a later stage when they're already in the process of dying, it'll be better to keep them alive for longer periods of time. And last thing that we have become a big fan of lately is small molecule or drug therapies. And we're trying to use various molecules to rescue the disease phenotypes using organoids and using mouse models. So that's what I do. And thank you very much again to Akshay. Thanks to Akshay and also to Manjari for the invitation, and to all of you for listening.
Thank you.
I'm going to stop sharing.
Thank you, Dr. Shweta and Dr. Anand, for enlightening us with starting with the basics of the retina and phototransduction, and then to the further extending our vision towards the therapy, what it can be used, and how the photoreceptors are so important to us for the vision. So I would like to everyone to ask question if there is any.
So,
Yes, Priya.
I'm Parul, postdoctoral fellow from Dr. Akshan and Slam. So, my question is little specific. I want to know the role of diet, especially fat diet, in the development of AMD pathology. Because recently I came across one paper in which microglia were accumulating lipid molecules, and these microglia were not able to phagocytize properly and hence causing neurodegeneration. So, is it the same case with the AMD or not?
You are asking me, I guess, right?
Yes, yes, sir.
Okay. Uh, AMD is a multifactorial disease. And when you say multifactorial means there are many, many different factors. No one factor. And that has been a problem in science. We have very reductionist approach. We try to sort of bring it down to the simplest level in order to figure out what is going on. Just remember, uh, AMD is an age related disease. It takes you 60, 70, 80 years. So your genes and genetic variants have been there with you for a long time. You have different kinds of diets that you are being exposed to over the years, right? And you have different uh, exposures, whether it's light, UV, some people smoke, some don't, and there are lots of different things are happening. So many times people say, oh, George Burns, I don't know if you know that person, but a very well-known old comedian. He lived over 100 years and he used to smoke and say, oh, but he was fine. Smoking doesn't hurt, but that's not true. Okay, so same thing is true with nutrition. When you say high fat diet, you will see that there are a lot of people who may eat different kind of diet yet don't get any coronary artery disease, but most people will. So AMD is along the way same thing. Don't go with just one paper. What they are looking at is in one model under one condition, under very severe circumstances, some phenotypes are being created. Okay. And so mice are a useful system in most cases, but mice do not have many features that happen in age-related macular degeneration. So you do not generate a phenotype of AMD in mice. So what you do is you have if a part of the phenotype, you have one of the what we call endophenotype that you observe. Yes, high fat diet is not good for retina. And lipids, there are many genetic variants in genes associated with lipid pathway which can have an impact on AMD pathology, and high fat diet is not going to be beneficial. It is going. But is that causing the AMD? It's contributing to AMD. How much? It depends on each individual. Okay. So what you are reading is one paper, and I'm giving you a very general answer. We can go very specific. You can send me the paper, but microglia is one part of it. There are many people who say it's an RPE defect. Some people who say it's a defect in choriocapillaris. Some people who say it's a defect in photoreceptor. But no, actually it's defect in multiple cell types and dysfunction. Actually, it's not a defect. I would say dysfunction of multiple different cell types and many different pathways which have to come together in a unique combination in order to give you the pathology of AMD. Not a single thing is going to do that.
Sir, my next question, sir, as you said, animal models are very specific and depicts, somewhat pathology or characteristics of a disease. So, what would be the best model? Is it the iPSC, patient-derived cells, which would be the best model for study any drug or any treatment? What would be that?
So, Parul, you're asking a very important but very broad question. If you look at the literature, iPSCs you derive from human patients can give you again one part of one part of the answer. Why? Because they are in vitro system. They're not in vivo. So mice are very good in vivo system, but they don't have the similar kind of features and pathology. iPSCs, on the other hand, are human derived, so they are much better to some extent. But on the other hand, here you're looking at uh, uh, one cell type or even the organoid, which is in, in the dish. Whereas in organism, eye is not in the dish. You are not creating, you don't have any capillaries there, you don't have any endothelial cells, you don't have. So what would be ideal? Ideal will be theoretically if you can make an eye in a dish and replicate. And there are people who are trying to reconstruct by using bioengineering protocols or by other methods some of these kind of features, but still you can reconstruct maybe choriocapillaries and RPE and those photoreceptors. At least you can bring a couple of these cells together, but you still don't have the whole eye. Means you don't go all the way to ganglion cells. So how the overall system is functioning is not going to be that simple, particularly for AMD being. And the aging is a big component. You do not you cannot have the aging phenotype in organ. But are organoids good? Yes, because they give you one answer. Are mouse models good? Yes, they give you another answer. So to understand AMD, you need to combine knowledge from multiple systems. Okay, so it's not for inherited Mendelian disease. It's a much simpler case, where is one gene defect in general and you have a phenotype. You can try, but even there, mouse models, even though theoretically they should be able to replicate or recapitulate the phenotype, they don't do that as well. Okay, the timing is different, and sometimes the phenotypic severity is different. So there are a lot of things that are unique to humans as an organism. What is the best model system? Humans. But you can't study human. But that is why we do human genetics. That's why we took human blood and post-mortem retina samples. That's why we do that. Why again? We take that knowledge, combine it with organoids, combine it with mouse models, combine it with yeast, combine it with fly. Each of these systems, in their own unique way, contribute to the overall understanding of disease, complex disease biology that we have. Does that sort of answer you? So I think you need to, as a scientist, we have to focus. We have to be reductionist because we cannot sort of work on all 20 things at the same time. That's why I was trying to show you that picture of an integrated or systems-based approach. We cannot do that so easily. I mean, it's just sort of beyond our capabilities, capability of a single individual. That's why we work in teams. That's why we have consortia. That's why we take knowledge from many different studies. Okay.
Thank you, Dr. Anand. I would like other our students to ask some questions to Dr. Shweta.
Namaste. Myself Kalyan. I am basically from the yoga background, yoga scholar under Dr. Akshay Anand. So my question is related to yoga. Actually, I want to ask you a research question which can be implemented for eye related to yoga. So basically in yoga, we are giving some eye exercise, and there is a kriya called Tatak Kriya. Basically, there we are stimulating our eyes through the candle light, and also we are giving some up and down, left and right movement of our eyeballs. So as Sir also told that he has also mentioned the yoga things to for the regeneration, means slow down the degeneration of the macula cell and also retinal cell. So how can we implement this for the eye study? Because there are very few, only very small amount of study we can see in yoga field related to eyes. So please suggest, sir, how can we implement this? Because if we are conveniencing to patient, or we are approaching the yogic way, so they are directly, so how can it will help? So can you suggest any idea how can we implement this and we can prove this scientifically?
I think I want Shweta to answer some questions as well. Shweta, do you want to take the answer that question?
Okay, I can try. I don't know exactly. So whatever you are saying is that you are just making some eye movements to, I mean, individual to do some eye movements, and you just want to see what effect does it have on retina. That's what, if you are asking, or you're asking me that how you can just take this project forward, or that's what I'm getting from you as a question. So see, yoga related studies are very less, I think, in the field of retina or in any field as such. So what you can do is you can recruit more individuals, and just to see before going for any like plasma or serum studies, you can just go for like what is the difference in the, is there any reduction in the ocular pressure, or things like that, that you can just see without any much interventions into the retina, because you can't just take human retinal samples or do sectioning over them. So you need some modalities that you can just uh, uh, do without any harm to the individual. So either you can take the blood samples or process them and then see that whether there are some changes in some markers of the retina. Of course, you can't be seeing any markers of retina in the blood, but you can see some changes, if any, in the serum samples. Or you can just, I think, first thing that come into mind is if you can just look for any changes in the ocular pressure, because you say that you are just asking them to rotate their eyes or look left and right or up and down. So I don't think that will create much of the difference as such. But these are the things that you can look for, and I don't know anything else for right now. Maybe if Dr. Anand wants to add into this.
So I think what I want to say is something very different, which is how to design a study. So Kalyan, what you're asking is a tough question that cannot be answered so easily. Okay. Whenever you design any kind of study, you need to have parameters for assay. Okay. You need to know what you're trying to look at. In a human being, when you're giving yoga or any exercise or nutrition or diet, those studies are very difficult in general. That's why not only yoga, but you remember these ARED studies that were done with vitamins are done with thousands of people and all that. And the reason for that is that humans in general have extensive variations in phenotypes. Whatever you want to study for eye function, you are going to see tons of variations among different individuals. How do you say that this change that you are seeing is because of yoga, right? Because every individual has different nutrition. Every individual lives in a different way. Some is rich, some is not so rich, some is having eats a high fat diet we were talking earlier, or low fat diet. Somebody is exercising, somebody is not, somebody is doing eye exercise in yoga. How can you say that the change that you're seeing, even whether it's in eye pressure or anything else that you're measuring, is caused by that one thing that you are doing? That is why those studies are extremely difficult. However, okay, I'm going to say, however, is it good for you? My philosophy in general is, if it's not bad for you and it doesn't hurt you, even if it may help you 2% or 5%, try it. Do I do yoga and I do pranayam, and I know the benefit because I can feel some of that benefit, but it takes years. So now these studies, that is why, have to be done in individuals, and you need to do a longitudinal study. If you want to design this has to be done in a you cannot compare a Kalyan to Shweta to Parul to me or to Akshay or to anybody. Why? Because we all are very unique. We have very different genetics. We have different food habits. We live in different environment. And you cannot control that. Therefore, for you to say that anything is happening in your eye because you did yoga exercise, eye yoga, it will be impossible. Nobody's going to believe you. I won't believe you, because you have too many. Why we study mice? Because we control everything that a mouse does, and we just change one thing in mouse, one gene here or one parameter. Then we can say, aha, that is caused because of this parameter, this suggestive of that, right? But in human, that's impossible to do. That's why, unless you teach yoga to mice and you can know the parameters, it's not going to be simple. Okay, so if you want to design this, one way to design that will be a longitudinal study, and that is, you take 10 individuals and follow them over a period of few years, and some with disease and some with no disease, and see if some of these things helped or hurt. Okay, so that's the design of study as far as I'm concerned. But basically, just doing yoga and saying that this change is because of this is incorrect in a scientific way, because there's so many other things happening.
Thank you, sir. Sir, one small question I want to ask. So, some practitioners are asking, there are so many patients are complaining that they are having the black spot, black circle around in front of them. They have some eye disorders. So, they are facing the problem with black circle in front of them, and also like spider web structure they are seeing in front of them, even though they are wearing specs. And also sometimes whenever they go to the sunlight, they are having the more secretion of tears. So what is the cause?
Okay, so I am not a clinician, so I'm not going to say anything to you. I cannot say, but I have been with a lot of doctors. So all I can tell you is that what you mentioned are many different problems. Okay, this is not one disease. So each person is having these different issues. The best way is go to, you're at PGI or go to your ophthalmologist. Specs is only to correct issues which are related to focus, okay, and ocular muscles to some extent, okay. But there are many other. Eye vision is a very complex process, as Shweta described, with so many cells. And I was telling, I have focused only on photoreceptors, but you have ganglion cells, you have optic nerve issues, you have inner retina, you have muscles, you have cornea, you have trabecular, you have so many things. So go to a good ophthalmologist. PGI ophthalmology doctors are amazing. Many of them are very good. So you just sort of, people have to go and take proper. They are not going. I mean, okay, I'm not going to say never, but yoga is not going to solve an acute problem. In general, our Indian medication, and I'm a big fan of even Ayurveda and yoga, but those things are more to maintain a healthy, good lifestyle so that you do not get the disease. But once you have the disease, it's an acute disease, it needs to be treated in a different way. Yes, nutrition is going to be helpful. I mean, there are many folks who I know in our own family who are MDs, and they decided to open these health clinics now, because a lot of issues that we as humans face are not just related to what we are seeing. Sometimes you can have eye problem which may be related to issues which are neurological. Sometimes there may be problems which are associated with different things. So I think maintaining a very healthy lifestyle is very, very important to prevent these things. So prevention is better than cure in most cases. But once you have it, you need to do something more serious about it. Okay, whether it is so once things are acute, then you may need to try. Okay, and that's why you need to go to an ophthalmologist and see what happens. And many other issues can be avoided by doing good things, by maintaining a healthy diet, by doing exercise, by doing yoga. And that's what I was trying to say. These things affect what we call, I didn't want to go there, but we have a big project now on aging and epigenome. And also we are beginning to, a lot of people are beginning to do microbiome. What you eat changes your gut microbiome, which can have an impact on many, many different facets in your body, including eye. That's why nutrition is important. Exercise, having good, peaceful mind, your blood pressure, so they're all integrated. We are a integrated system. A human body, eye is not existing alone in this thing. It's connected to everything. So I can't answer your clinical questions, but all I can tell you is that they need to go to doctor. I need to go, actually. It's already getting very late. I told Akshay that I'm going to be there for 10 more minutes, 15, but it's already, I am actually supposed to be attending a meeting.
Yes, sir. Sure, sir. You can leave now. We will be ending our session.
Okay. Thank you very much.
Thank you,
Okay.
sir.
Thank you. Thanks, Akshay. Thank you. Thanks, all of you guys.
Thank you, sir. Thank you. And I'll be telling you that there are 2 questions related to yoga, that which yoga can help, that has been already answered by Kalyan. So we will be joining you tomorrow also with a talk on quantification on immunohistochemical images by Dr. Abhijit Banik. So see you tomorrow at 6 p.m. Thank you.